Hello, my name is Sadie Shelton. I am going to be doing a presentation for case connections number 2. First, I'd like to start off by talking about the first case, which is about pyruvate kinase. Pyruvate kinase is an enzyme that is involved in glycolysis. Its starting product is phosphoenolpyruvate, and its end product is pyruvate, which is then turned into acetyl-CoA and used in various other biological processes, such as can be turned into lactate, can be used in the Krebs cycle, can be used in a lot of other things. But for right now, we're just going to talk about phosphoenolpyruvate turns into pyruvate through pyruvate kinase. This also involves the phosphorylation of ADP into ATP. So this is one of the energy producing steps of glycolysis. So there are a couple things that regulate pyruvate kinase. There are some that inhibit it, and then there are some that keep it going. So some of the things that encourage pyruvate kinase are AMP. So AMP is going to encourage pyruvate kinase to continue making pyruvate because you need ADP. So if you have a lot of ADP that needs to be turned into ATP, what is a precursor of ADP but AMP? So if you have a lot of AMP, this needs to be turned into ATP, and one of the ways that that can happen is through the use of pyruvate kinase. So if you have a lot of AMP, you're going to be using pyruvate kinase. Pyruvate kinase is also one of the enzymes in glycolysis that uses feed-forward activation. So if you go back a couple steps in glycolysis, you create fructose 1,6-bisphosphate. So if you have a lot of fructose 1,6-bisphosphate, you're going to need to turn this into pyruvate to use it into the Krebs cycle. So if you have a lot of if you have a lot of fructose 1,6-bisphosphate, that is going to encourage pyruvate kinase to because eventually fructose 1,6-bisphosphate turns into phosphoenolpyruvate, and once you have a lot of that substrate, you need to turn it into its product using pyruvate kinase. So it's going to encourage pyruvate kinase to work. Some inhibition of pyruvate kinase is going to be ATP. If you have too much ATP, then you do not need glycolysis to create more ATP or the breakdown of glucose. This is going to stop glycolysis, which is going to stop pyruvate kinase. So if you have a buildup of any of these products that pyruvate kinase makes, so ATP, pyruvate eventually turns into acetyl-CoA. So if you have a buildup of too much acetyl-CoA, that's going to then come back and inhibit pyruvate kinase because the body is all about homeostasis and balance. So if you have too much of the substrates, you're going to encourage pyruvate kinase. If you have too much of the products, such as ATP or acetyl-CoA, then that is going to inhibit pyruvate kinase. Alanine, interestingly enough, is also an inhibitor of pyruvate kinase. Pyruvate, through the process of transamination, can turn into alanine. So of course, if you have too much pyruvate, that can turn into too much alanine through transamination. That is going to come back and inhibit pyruvate kinase. So next, I was given a scenario about the different types of pyruvate kinase, and there's one called M2. I think the M is for muscles, and then it's involved in the promotion of tumor growth and cancer. So upon my research with pyruvate kinase M2, of course it can be used to promote aerobic glycolysis, but it also has another feature as to where it can be there's like dimers, so through dimerization it can be split into and activated into sort of a protein kinase function. So instead of turning phosphoenolpyruvate into pyruvate, it turns into this completely different function, into this protein kinase, and also transcription factor. So the protein kinase and the transcription factor help promote tumor angiogenesis and tumor growth. The transcription role allows for it to activate certain genes in the nucleus, and this helps synthesize raw materials for the daughter cells of the cancer cells. So it helps create nucleotides, amino acids, and phospholipids. All of these things can have parts of glucose, and the nucleotides have a glucose backbone, and so the pyruvate kinase M2 can take the glucose, and instead of using it for glycolysis, it can divert it into creating these materials for these cancer cells. Tumor growth is also really fast-paced, and it metastasizes and it grows quickly, and so it's going to be using regardless of the oxygen or glucose cells. I read that pyruvate kinase can also turn into this protein kinase because these cancer cells are going to use kind of more of an anaerobic glycolysis, so they're going to do pyruvate to lactate, and you could get pyruvate through alanine, or oxaloacetate can turn into pyruvate. So if you have pyruvate from, if you make pyruvate from things other than glucose, you're going to use it in anaerobic ways, so it's going to turn into lactate. So this increase in lactate turns it into a highly acidic environment, which is good for the tumor cells and allows them to metastasize and have really fast-paced growth. So the reason I think that pyruvate kinase M2 kind of diverts away from aerobic glycolysis is it turns into this more of a protein kinase and transcription factor, and diverts the glucose from being used in glycolysis and helps create other factors such as the nucleotides, amino acids, phospholipids. It helps create more products for the daughter cells to grow quickly, and then the protein kinase also encourages the angiogenesis and tumor growth. So that was for the first case. The second case is all about glycogen phosphorylase. So I am talking to a patient that just found out that they have a glycogen phosphorylase deficiency, and I'm going to explain to them how glycogen phosphorylase works. So obviously, this is kind of a complicated chart. So what I'm just going to kind of explain to you the most simplest way that glycogen phosphorylase works. So we have in our skeletal muscles, we have receptors that release calcium when we contract it. So when we move our muscles, we kind of bend them out and in like this. Any sort of like muscle movement releases calcium. This calcium activates kind of this cascade. It activates a bunch of different enzymes that activate one another. So it kind of goes from calcium to protein kinase A to CAMP. Actually, it's flipped. So it goes calcium, protein kinase A, CAMP, and this activates phosphorylase kinase, which then phosphorylates glycogen phosphorylase. So basically, you just need to know that calcium activates all of these different enzymes, and then we get glycogen phosphorylase. This, usually when it works, will take a glycogen, which is a stormed form of glucose, and break it down so we can release glucose so it can be used to create ATP, and ATP is energy. So when we have a deficiency, the calcium will increase in the cell because it breaks down, it's released, the muscles contract, it releases the calcium, but this glycogen phosphorylase can't break down the glycogen. When we can't break down glycogen, we have no free glucose, which means there is no way for the muscles to create energy, and we get what's called the ATP shortage. And that's when, because the ATP shortage, there isn't enough energy for the muscles to work, until they break down and they start to hurt really bad, which is what happens when you exercise. And that's how the doctors were able to figure out you had this glycogen phosphorylase deficiency. So basically, when you move your muscles aren't able to create energy to help them keep moving, which is why it hurts when you exercise. So I see you also had some questions for me about kind of what you can and can't do. So interestingly enough, you can do long walks. So the types of exercise that is really going to worsen your condition is going to be really hard, strenuous aerobic exercises. So if you're doing like lots of running up and down hills, if you're doing really long, hard bike rides, that is going to use up a lot of ATP, and it's going to really make your muscles hurt because there's not, the glycogen phosphorylase isn't working hard enough to be able, or isn't working at all really, to create that ATP. So you want to avoid really hard, strenuous exercise. Also like really fast anaerobic intense exercises, so like HIIT works, any sort of stuff like that isn't going to work very well. But a long walk, as long as it's, say you're kind of walking down a path, as long as there's not a lot of hills, you're not like running really hard, just kind of a nice gentle walk, that should be okay. Obviously, if it starts to hurt, stop, take a break, don't do that. But long walks with your friends should be okay. Next, you have questions if you're going to need to get up in the middle of the night and eat. So interestingly enough, glycogen breakdown in your muscles is not your blood glucose dependent. So you have two hormones that kind of facilitate your blood glucose. You have insulin, which of course, if your blood glucose is too high, it's going to take the glucose and turn it into the glycogen. But if your blood glucose is too low, you use the hormone glucagon, which is going to break down the glycogen. So glucagon only affects the glycogen in the liver, but your condition affects the glycogen phosphorylase in your muscles, which is not glucagon dependent. So thankfully, you are not going to need to get up in the middle of the night to eat to help raise your blood glucose, anything like that. Your last question is, you're worried, of course, about a high lactate buildup. So as we talked about, intense anaerobic exercise is not the best idea. You're not going to want to do that. It's going to hurt your muscles. You're not going to be able to make enough ATP to give enough oxygen to your muscles. So interestingly enough, glycogen turns into glucose, and then that glucose can be turned into lactate in sort of an anaerobic glycolysis. But if we're not able to access that glucose in the first place, if we are not able to break down that glycogen in the first place through your glycogen phosphorylase, you're not going to be able to make lactate. So you're not going to worry about a lactate buildup because you're not going to be able to make enough lactate in the first place because you're not going to be able to break down that glycogen. I hope that I was able to answer all those questions. And of course, if you have any more, you can definitely ask them and I'll answer those for you.